All right, kids, here we go. This one is going to be on meiosis. So remember, meiosis is the process of making gametes or sex cells. So we're going to kind of be all over the place today. I'm going to be going back and forth between um, this slideshow and then another one that's got the paper dolls. If you remember those paper dolls that we were working with for mitosis? We're going to be using those again, and so I'm going to walk you through the stages of meiosis using those. So you might have those out. Make sure you have your guided notes sheet if you need to add stuff to that. And this will be kind of a review for you in case you either missed class or you just need to brush up on what we talked about already. So this first question I posed to you guys said, where do your chromosomes come from? So think about that for a second. And hopefully you said they come from your mom and your dad. Your mom and your dad had to reproduce to make you. Yes, that's the uncomfortable truth that at some point in the past they did have sex, but that's where you came from. And you got your chromosomes from them. Remember, you get half from mom and half from dad. So this funny little animation here, when I click in my smartboard for reproduction, this is what happens. So apparently, if you hold hands with somebody of the opposite sex, a baby will crawl across the floor in front of you, and that's how reproduction happens. Anyways, funny animation, but babies come from a mother and a father, which you already knew that. So for this baby, if he got, a chromosome that looks like this from dad, he's also going to get one that looks like that from mom. Notice how they're the same pattern here. They're the exact same chromosomes. Now, we already talked about alleles and how there's different forms of genes that are on these chromosomes. I mean, mom might have an allele for, you know, one hair color here, and dad might have an allele for a different hair color here, but they code for the same traits. Okay, so then again, if you get a chromosome that looks like this from dad, you're going to get one that looks like that from mom. So this is those chromosome pairs we were talking about. If you remember, we said that humans have a total of 46 chromosomes, which equals, 23 pairs. So this is what I was talking about with pairs. So for each one that looks like this, and let's just call it the first chromosome, you're also going to get a first chromosome from mom. If this one was the second chromosome, you'd also get a second chromosome from mom. See how that works? So all together you'll have 46, but there's just 23 pairs. All right, and so we call those chromosomes that are the same, we call them homologous chromosomes. Homo, remember, we've talked about these prefixes many times. Homo means the same. So this chromosome and this chromosome, they're homologous because they're the same. Likewise, these two longer ones, these would be homologous chromosomes because they're the same. So a few little things here. They code for the same genes. You get one from mom, one from dad, and humans have 23 pairs. Here, this is just a little diagram that shows that. This is called let me move this guy out of the way here. This is called a karyotype. We talked about them briefly in the past. So this is a karyotype, and you can see here, there's 23 different pairs, the 23rd pair being our sex chromosomes down here. And so if we look at this one, an X and a Y, that'd be a male. So this is a male's karyotype because they have an X and a Y chromosome. But you can see for the first chromosome, they've got 2. The second chromosome, they've got 2. They look the same. If you counted up each individual chromosome, you'd have 46. So if a male sperm cell combines the DNA with a female egg cell, what happens to the number of chromosomes in the offspring? Well, you can see where this would become a problem. If egg and sperm both carried 46, so 46 plus 46, that means that each time we reproduce, we'd be doubling the number of chromosomes in the cell. That is a problem. We don't want that to happen because we'll see a little bit later in this unit that when you have more chromosomes than you're supposed to, bad things can happen. So we don't want that. That's bad. So how are we going to end up with a child who has 46 chromosomes? Well, the obvious way is that we're going to have half of the normal amount of chromosomes in each egg and sperm. And this is where meiosis comes in. Remember, mitosis made identical daughter cells when the cell reproduced. In meiosis, we're going to be making cells that have half the number of chromosomes, so 23 instead of 46. So a little bit of terminology here. This word diploid, remember di, that means 2. Haploid here, kind of sounds like half. That's going to be half of diploid. Well, we say that diploid is 2n. Okay, well, what on earth does that mean? The n refers to the number of types of chromosomes. So, for example, humans, let me finish writing this here. Humans have 46 total chromosomes, but we have 23 types or 23 pairs. So for humans, our n number equals 23. Now, n number can be different for different species. For example, there's some that have, over 100 different types of chromosomes. There's some that only have 7. I think um, Drosophila, which is a fly, I think might have 7. I'd have to look that up. But the number of types of chromosomes they have is can be different for each species. So humans, our N number is 23. We have 23 types. You get one from mom and one from dad. That's why this 2 up here means I've got 2 of each of those 23 types. So that means diploid. 2 of each type would be diploid. Most of the cells in your body are diploid. Haploid means that you only have N chromosomes. So in this case, 23, because humans have 23 different types of chromosomes. So notice you only have 1/2 of each pair. This is what we see in egg and sperm, which is very, very useful because think about it. If we add an egg and a sperm together, 23 plus 23 gets us back up to 46 total, or 2n. And that's what all the cells in your body have, like your skin cells, your brain cells, your muscle cells. They're 2n. They've got 2 of each type of chromosome. This is what most of our cells are. This is only found in sex cells. So in overall meiosis, the goal is to cut the number of chromosomes per cell in half. So instead of making daughter cells that have the same number of chromosomes as the parents, we make daughter cells that have half the number of chromosomes as the parents. They still have one of each type of chromosome, but only one. So what we end up doing then is separating those homologous chromosomes. So if we back up real quick, remember these are homologous chromosomes, like this one and this one are here. Maybe this is easier. Those are homologous chromosomes. They're the same kind. They're the first chromosome. One of these came from mom, one came from dad. Well, in order to make sure that the egg only has half, or 23 chromosomes, we're going to split apart each of these pairs so that you only get one in each egg or sperm. So we'll take a look at what that looks like in just a second. And this ultimately leads to making sex cells, which are called gametes. Gametes is just a vocabulary term you need to know that means sex cells. That could be egg or sperm. All right, so this is a big diagram. This is showing us what happens during meiosis, and we're going to walk through all these phases together. If you look, we recognize these. Interphase, there's G1S, and G2 should be in here too, but they didn't show it because it looks the same as this. And then we go through prophase, prophase 1, metaphase 1, anaphase 1, telophase 1, and then we go to prophase 2, metaphase 2, anaphase 2, telophase 2, and we ultimately end up with 4 different gametes. Now, what I want you to notice is this right here. Meiosis is not a cycle. These gametes are not going to start dividing again. Their goal is to join with another gamete. So like if this was an egg, it'd want to join with a sperm in order to make a zygote. That just means, a fertilized egg that's going to become, another individual. So it starts with a normal cell, ends up with these gamete cells. They do not start the process over again. Okay, so meiosis is not a cycle like mitosis is. Now there's some things that should look familiar here, but there's some key differences too that we'll talk about as we go. All right, so this is just showing you what we're going to do. We're going to go through meiosis 1, which is prophase, metaphase, anaphase, telophase 1, and then meiosis 2, prophase, metaphase, anaphase, telophase 2. Okay, that's really the only reason I'm showing you this is just so you can see it. All right, so let me hide some of this so you're not super distracted here. Oh, where'd my little toys go? We'll put a cell shade up here. And oh, what happened to my, oh, there we go. Haha. All right, let's go back to this. So the major parts in prophase 1, we have the formation of these things called tetrads. So these are the homologous chromosomes paired and with their sister chromatids. Okay, so let's take a look at what this means. Let me make a new slide here. Remember we talked about sister chromatids before. So let's just use we'll use red for mom and blue for dad. So let's say this is one of your chromosomes. Okay, it doesn't matter what chromosome it is. There's a chromosome. There's the one that you got from mom, and here's the one. Oops. Here's the one that you got from dad. Okay, we'll leave this guy for now. During interphase, remember we have to make a copy of all of these. So here we're going to make a copy of the one that you got from dad, and we're going to make a copy of the one that you got from mom. Oh, bother. Now, because these are exact same copies, do you remember what we called these? We called them sister chromatids. Okay, so these are sister chromatids because they're the exact same chromosome. They're the dad version, the original and the copy. And they're joined together, if we remember, this thing in the middle that joins them together, that's called a centromere. Now, normally, we are just concerned with these guys, and that's it. Oops, didn't grab him. But during prophase, during meiosis, this pair of sister chromatids and this pair of sister chromatids join together because this chromosome is the same type as this. It's just this one came from dad, this one came from mom. These two are homologous chromosomes. Okay, so this and this, these are homologous. And what happens is they are going to clump together in something called, if we look at all four of these together, so the two homologous chromosomes, that'd be red and blue, and their sister chromatids, so the two reds and the two blues, they form something called a tetrad. Now, you may recognize this prefix tetra. That means four. That should be obvious why it's four. Okay, so that's what I was talking about with tetrads. So if we look here, we form tetrads. That's just the homologous chromosomes and their sister chromatids joining together. We also get this little thing happening called crossing over. Now this is really kind of neat. What happens is, see these two, how they're kind of crossed right here? So you've got this sister chromatid and this sister chromatid, they're crossed with each other. What can actually happen is, let's see if I can do this here. Need my eraser tool. Oh, here it is. I'll get rid of you. We're going to erase these. What happens is a little bit of the DNA from the mom chromosome gets hooked onto the dad chromosome, and vice versa. A little bit from the dad chromosome gets hooked onto the mom chromosome, and that's called crossing over. Now I want you to think about why that might be good. Okay, think about each of these individual chromatids now. There's four of them there. Are they all the same anymore? So what do you think the benefit of that crossing over is? Think about that. That'll be something that might be, like a test type question. All right, so we're going to have our homologous chromosomes paired. That's forming a tetrad, and we get crossing over. That's when we switch parts of the chromatids from the mom and the dad versions. All right, so let's look and see what this actually looks like. So here we are. We're using our paper dolls. Notice we're in prophase, so our nuclear envelope should be disappearing. And here I've got these sister chromatids, because they're the mom version, and these sister chromatids, they're the dad version. Together, this whole thing is called a tetrad. Okay, so that's during prophase. So really, when we looked at mitosis, we'd see these guys, off by themselves. But during prophase and meiosis, they're up there together. Okay, so this group of four is a tetrad. There's a tetrad. There's a tetrad. These two blue, they're sister chromatids. These two red are sister chromatids. The blues and the reds together make up homologous chromosomes, okay, because they're the same type. After that comes metaphase 1. Now remember, we know that during metaphase, things tend to line up in the middle. So here, instead of having our sister chromatids line up in the middle, the tetrads line up in the middle. So let's go ahead and take a look to see what that looks like over here. So we're going to switch to the next slide here. So here's our metaphase 1 during meiosis. Notice, ooh, I forgot I did that. See, we've got some crossing over that happened there. So here we have our tetrads lined up towards the middle. Notice the orientation, that's very important. They're lined up this way, not this way, okay? So what do you think is going to happen to these tetrads in the next phase? Think about that. In mitosis, we would split apart the sister chromatids. But think about what's going to happen based on the way they're set up here. Just as a reminder, these are called spindle fibers coming out from the centrioles. They're going to help pull things apart. So as we go to the next phase, anaphase, here's the big difference between meiosis and mitosis. During anaphase 1, our homologous chromosomes are separated. In other words, we break apart our tetrads. From this point on, because we're separating our homologous chromosomes, we're not going to be diploid anymore, okay? We're not going to be this 2 of each type in each cell. We're going to go to haploid. So here, we're separating all of these homologous chromosomes from each other. So one's going to go to one cell, one's going to go to the other cell. So from this point on, we are not like a normal cell anymore. We're not diploid, we're haploid. So let's take a look at what that looks like over here. So watch closely. Bam. We separate our homologous chromosomes. Notice the sister chromatids are still together. Okay, these are sister chromatids. But our homologous chromosomes, so the mom version of B, the dad version of B, they're separated from each other, and this is eventually going to divide. So even though there's 2 chromosomes here, they're both B chromosomes. They don't have a different version of the B chromosome. Okay, so that's really important. All right, after anaphase 1 comes telophase 1, and that's just where the first 2 cells divide and prepare for meiosis 2. So let's take a look. Here's telophase 1. So notice the cell membrane is starting to divide those 2 apart. All right, now normally during telophase we'd get our nuclear envelopes reappearing, but since this is going to start another round of division, they don't really reform. So after telophase 1, we have prophase 2. So this is the second round of division. From here on out, we're haploid cells. Okay, remember they have half the number of chromosomes they started with. Now that can be confusing when you look at this because if you look, there's 1, 2, 3, 4, 5, 6. But you've got 2 Bs that are both dad versions, 2 A's that are dad versions, 2 C's that are mom versions. And it doesn't really matter which way these went. It's completely random when they line up at the middle. So I could have all blues over here and all reds over here. But the point is, for each type of chromosomes, like B or C, they only have one version. In this case, they have the dad version of the blue for B and the mom version for C. So they only have one version of each chromosome. Yeah, there's 2 of them there, but since these are sister chromatids, they're exactly the same. Doesn't count. So it just gets ready to divide again. So metaphase 2, our sister chromatids now are going to line up in the middle. So this is what usually happened during metaphase, during mitosis. So here we go. This is metaphase 2. Notice the sister chromatids are lined up in the middle of each cell. So this dark line means these are separated. We've got 2 separate cells going on here. So what do you think is going to happen to these sister chromatid pairs in the next phase? Think about that. And here's the answer. During anaphase 2, now we finally split the sister chromatids. Okay, we split the sister chromatids during anaphase of mitosis, but it's not until anaphase 2 of meiosis that the sister chromatids split. So here's anaphase 2. So now you can kind of start to see we're going to have 4 different cells that are being made at the end of meiosis. And then finally, we've got telophase 2. And so those completely separate. So we end up with 4 daughter cells that are haploid. So if we go back to our models here, notice the nuclear envelope's reforming, the cell membrane is forming. So at the very, very end, this is what we have. Now I want you to look in each of these. So let's just take this one for example. There's a blue chromosome a blue chromosome B, with a little bit of red on it from crossing over, and a red chromosome C. There's 1, 2, 3 chromosomes in this cell. Compare that to what we started with. 1, 2, 3, 4, 5, 6. What's 3 compared to 6? Half. So these are all haploid cells, or what we would call N, because they only have 1 of each type of chromosome. Now again, I'm just doing a made-up organism here. If we did this with humans, there'd be 23 chromosomes in here with when they're making gametes because they have an N number. But I just did this because it's a lot easier to see with just 3. But the point is, at the end of meiosis, we only have half of the number of chromosomes that we started with. So we go from 2N to N. All right, and that pretty much wraps it up. So I'm just going to bring us back to this diagram and just show you here. We start out with, look how many chromosomes there are here. There's 1, 2, 3, 4. There's these would be homologous, and these are homologous. And then over here, these are sister chromatids that are homologous to these sister chromatids. So watch what happens as we go to prophase. They're going to join up. They make those tetrads. Crossing over can happen. During metaphase 1, our tetrads line up. And here, our tetrads are split. So from this point on, our homologous chromosomes are separated. So at this point right here, so during anaphase 1, we go from being 2N to just N. After that point, this is going to completely divide. And here we're going to we're going to go through another round of division. Here we've got the sister chromatids lining up in the middle during metaphase 2. During anaphase 2, the sister chromatids split apart. And keep in mind, these are the exact same. So we're still just N value right here. We're not 2N. And then these are going to completely separate so that we end up with 4 haploid or N either egg or sperm cells. Compare this to this. 2 chromosomes versus 1, 2, 3, 4 chromosomes. That's the goal of meiosis.